speaker is Professor Ajit Kumar Saxena, Professor and Head, Department of Pathology, Lab Medicine, All India Institute of Medical Sciences, Patna, India. And the topic of talk is Exploitation of Circulating Tumor Cells to Mitigate the Challenges in Early Diagnosis of Breast Cancer Patients.
Good afternoon, and respected chairpersons. I am sharing my work, what I am working presently on early diagnosis of the breast cancer patients. What I feel, whether the diagnosis is more important or the treatment is more important of the cancer patients. We have a several time discussion with Dr. Gopinathan, who is sitting here, and he certainly advised me also when I meet in Bangalore. And Dr. Sinha is also here from US. So I am trying to improve this technology for early diagnosis. If we get an interesting result, we will do some devices for the cancer diagnosis with special reference to the cancer. So how the cancer develops, who is responsible behind it? We are responsible for the development of the cancer as the evolution of human beings takes place, and we started off playing the modern gaskets. So these are the very common figures who is suffering from the breast cancer in India and abroad and USA, and she is an actor. And Angelina Jolie, she removed the breast both when the mother was BRCA1 and BRCA2 positive. And the Manisha Koirala, she's a well-known figure of my country, now they are surviving. So the cancer is not a death sentence what people think. So this is the normal anatomy of the breast cancer, and which of the more prone tissues suffering from oncogene activation is the blood-rich area of the areola. So no doubt that the cancer is stimulated by the genes, group of genes, and the genes is influenced by the epigenetic factors. We have a oncogenes, but we are not suffering. It means there are certain environmental factors, and that is influenced by the environment. And that molecules are mutagenic in nature, our dietary factors, and even though some races are also being reported for exposed with the physical environments. So, and the most important aspect and characteristic feature of the cancer cells, they show genetic heterogeneity after mutations. So the complexity of the signaling is very, very complex, because we still don't know that the stem cells play a very important role mediated P53, or P53 is mediated stem cell like OCT4, Nanong, SOX2 on the right hand side. On the left hand side, there is a P53 and mediated signaling, and epithelial mesenchymal transmissions marker EMT. And this is one of the most complex, and the clinicians are trying to reverse the transmission from mesenchymal to epithelials for the improvement of the cancer patient's life. How to improve? There is only one way for the early diagnosis based on genetic screening, and that help for the diagnosis. And you have to decide whether the disease is a familial or sporadic. It means there is maybe a BRCA1 or BRCA2 positive for the germline mutation or the soft light mutations. Maybe, and that will help for the early diagnosis and better management for the cancer patients, especially breast cancer. So the cancer diagnosis and the treatment is first comes to the symptoms. You have to contact to the doctors timely, diagnosis, and start early treatments. Failing which, you have to decide the suffer. So the cancer diagnosis is based both invasive and non-invasive. Our technique is based on minimal non-invasive techniques based on the circulating tumor cells, the frontier in cancer medicines. Because there is a tendency from primary tumors, they shedding the cells and come into the blood streams and they are moving. So the study of circulating tumor cells is highly complex in nature because in total cell population is vary from 1 to 2%. And after that, you have to characterize from the epithelial mesenchymal transitions marker, the SOX4, the first transcription factor, and then epithelial cell adhesive molecules, and then CK19 when the cells are enter into the matrix and during invasion. So it is very difficult. The earlier scientists reports that we collect the circulating tumor cells from 10 to 20 ml of the blood samples. What I feel it is against the ethics. So what I have done with the help of biotechnology and tissue engineering, we have the origin of secondary tumorigenesis is also very important because they are circulating in the bloodstream and they may be enter into any of the neighboring tissue like the liver and the kidney, lungs, and the other organs. And once the epithelial cells become activated, they are very aggressive in nature and they act as a mesenchymal activities. So that's why the epithelial cells change their morphology. So what we have planned with the help of biotechnology, we have minimum amount 0.05 ml of few drops of the bloods were taken out and we have cultured. And we have tested the genetic heterogeneity by using RT-PCR and flow cytometry analysis. Second factor comes how to determine the risk factor by studying the MTHFR C677T gene polymorphisms. Because you must be knowing the cancer cells are immortal cells and they are growing continuously, but there is a need of the food supply or DNA methylation through folate metabolisms. But a day will come, the cell die and the individual dies due to the defect in the folate metabolisms. So this is by sketch diagrams where we have cultured the cells in sterile CO2 incubators and we have isolated the cells with the help of common Ficol gradient methods after 72 hours, and we compare with gradient cultured and non-cultured cells. And this methodology we have patented, and we have cell populations, epichem populations by using monoclonal antibodies were calculated by the flow cytometry analysis, and we have done by incorporation of the 5-hydroxycytidine, which is one of the most common drugs. And we have seen, if the histogram, you will see the 99.27% cell population are positives. In one patient, 1630, and the another patient, you will see the histogram, the cell populations, epichem positive cell populations was decreased 75%. This clearly showing that the two individuals or two patients have showing different genetic susceptibility toward the anti-cancer 5-hydroxytidine in breast cancer patients. As a model we have used. Then we have used another marker of the transcription factor, SOX4. This is one of the most important transcription factor after activation of the oncogenes KRAS that in different culture conditions, if you look the first bar diagram figure, that you will show that the control groups and then systematically the cell count or the cell populations are decreases of the SOX4 by using monoclonal antibodies. But third and fourth, when we use a cyclophosphamide, that there is not a significant difference in the cell populations. And we have also used a mutagens because there might be some environmental factor of the mutagens because eastern part of the countries are very rich in arsenic exposure, a very high content of the arsenic in the blood. If it is exposed with the arsenic, that the whole food chain will be affected because not animals, the vegetables and the human beings too. I am moving a little bit fast. I will give only 1 minute more to conclude it. And this is a SOX4 in CTC in cell populations by, you can see from control and the rest of the groups, these cell populations is down-regulated. You can see the aggressiveness of the circulating tumor cells, that when the epithelial molecules is either absent, mutated, or overexpression or down-expression, they behave like the aggressiveness and they change their anatomy. These are the single stem cells, circulating tumor cells. You can see the beautiful of different colors in the microscope. We have taken a photograph of my student and the chromatin material by the arrowhead. And these are the expression of the EMT marker in circulating tumor cells, both from the biopsy sample and compare with the blood samples. You will see the arrowhead. There is a complete either mutations of the EMT marker or down regulation or up regulations. But at compared to when we compare the blood samples, there is a more prominent as comparison to tissue. The Vimentin is a marker of the metastasis, and this is the only marker which showing the isoforms. We didn't know why the isoforms of the Vimentin is available, but it is predictive that it might be bind with the other signaling molecules. You can see the arrowhead either missing, or in different patients, the isoform of different expression is there. So recently we have published this article in Genetics and Molecular Research of 92.3 impact factor, more than nature of the medicine and biotechnology. And before you come to the molecular genetics, you have to be use the classical genetics. We should not forget that how does it look your human genome in the form of a thread, the chromosomes. So we have developed a beautiful karyotypes. You will see the arrowhead, there is the structural and numerical variations, and between homologous and heterozygous in conditions. And polyploidy is one of the characteristic feature of the malignant cell when the normal is transformed in the malignant. So there is a lot of aneuploidy there, trisomy 21, and double minutes. Double minutes, it is believed that the euchromatic extra chromosomal expression is there. So this is the expression of stem cell marker in pancreatic tumors. I am moving a little bit fast to share my work, and the pancreatic tumor with MTHFR gene polymorphism to determine risk factor. This is also published. This is a CTC in liver cancer. By using RT-PCR, you can see there is a SOX4 is completely expression is missing by the arrowhead. And this is a KRAS mutations in CTC of hepatocellular carcinoma. And in acute lymphoblastic carcinoma, there is a karyotype is a very important aspect to diagnose. That is the extra copy of chromosome 21 is there arrowhead and confirmed by the FISH analysis. This is the SOX4, epicam, and CK19 in acute lymphoblastic anemia. The finding was published in Cancer Medicine, and the MTHFR C6 double polymorphism risk factor in circulating tumor cell in LLL. When we have studies about any of the EMT markers, we simultaneously also study the risk factor by using MTHFR C677T allele. So now the miRNA expression help for the clinical diagnosis and for the signature. The unique miRNA signaling in tumor can be helped, and variety of the signaling is there. This is a bioinformatics tools. We have found it and predicted that the miRNA out of 3 species around 205, 155, and miRNA 25 could be used not only as a biomarker for the early detection, but it could be also used for therapeutics. And most of the in USA as a clinicians they were using. So in conclusion, the cytogenetics useful tactics for evaluate the frequency of the complex chromosome rearrangements in heterogeneous cell populations. Gain and loss of chromosome is a characteristic feature for genetic susceptibility for the individuals. The prognostic significance of the chromosomes and loss of heterozygosity of chromosome 11 of different regions and loss become relevant for the early diagnosis. So DNA copy number help is a genetic susceptibility for individuals to individuals vary. because the clinicians, if they prescribe the drugs, one patient become healthy, is curable, and another patient of the same therapeutics, they are unhappy and they return back it. So the clinicians become confused that we have given the same drug of the same symptoms, but why not working? It means there is a need of personalized medicines. And that personalized medicine is based on the genetic susceptibility. And the MTHFR gene polymorphism determine the risk factor in the cancer patient on the basis of 3 genotypes in homozygous CC and heterozygous condition CT. And these are the heterozygosity of the breast cancer patient, and this is beautiful photograph by using RT-PCR where all the 3 genotypes are available. The green one is rare homozygous conditions, and rest red one is a heterozygous condition where 3 bands were there. And the gallbladders, the pancreatic tumors, and the ovarian cancer. We have done a variety of, and this is Williams tumor. Williams tumor is a rare disease of the pediatric age groups. And the CTC and miRNA offers a promising target for early detection, prognosis, and therapeutic for the cancer, and scope of developing new strategy based on gene-protein drug interaction, and for pharmacogenomics and personalized medicines. Acknowledgement, our directors and my research team, Dr. Vijayendra, he is MCH in pediatric surgery. Pitanjali is a chemotherapeutics. Dr. Meenakshi is a postdoc, and the Dr. Veena, she is also surgeon, and Dr. Manoj, she is also surgeon. I would also like to thanks to my patients, and this is my academic contribution, who published the book from the Springers and research book, Basic and Clinical Consequences of Genetic from Cambridge, and first book, Gene and Male Infertility. And my lab was inaugurated by Padma Shri Father of DNA fingerprinting, Dr. late Lalji Singh. And one of my negative and hindrance factor is the small baby who always come into the lab and care to log the lab. And thank you very much for the audience.